Okay folks, I'm going to do my best to make this screencast to do a demonstration on how to do dihybrid cross. So remember, a dihybrid cross is when we're looking at two different traits, two different varieties of two different traits. So for example, in this particular example, I'm going to look at height. So we're looking at height here. That's one trait, and they can either be tall or short, so that's a variety of height. And then the other one is going to be the color of the pea. And in this case, we have two varieties. They can either be yellow or they can be green. And we're talking about complete dominance right now, which means if you have the dominant gene, that is all that's going to show up. Alright, so we've got height and we have color as our two traits. Since there are two traits, that's a dihybrid cross. Monohybrid cross would be if we just looked at one. So in this particular example, if I gave you a cross, and usually I'll give you a story problem to make you figure it out, but for this I just want you to look at how we would do it. If we crossed two heterozygous plants, so they're heterozygous, which means they have one of each of the genes, and they're tall and yellow. So they're both showing the dominant phenotype. So the cross looks something like this. It has one of each of the tall or height genes, the tall and the short, and it has one of each of the color genes, the yellow and the green. And both parents are the same genotype. They are heterozygotes. So how we do this, and how I have always done it, is I take the genes, or I take the genes that are available for each parent, and I separate them by commas, here, 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 and here. And then what we need to do is we need to figure out the possible combinations that you can get of the gametes. And remember, the gametes are the sex cells, so the sperm or the egg. Alright, now when we're doing a cross like this, it doesn't matter which one is the mom, which one is the dad, which are sperm, which are egg. All we need to figure out is what are the possible ways that these T's and Y's can form a gamete. So I use the FOIL method, which you do in your algebra classes, the first, outside, inside, last. So we will look at one parent at a time. So for this one, we're going to start on the left side, and we're going to put the gametes over here. So if we use the FOIL method, then what we look at is the first two letters in each set, or each parenthesis set, which in this case is the big T and the big Y. Alright, so that's the first possible combination of genes that a sex cell or a gamete could have. So we put a big T over on this side and a big Y there. Now, the next letter in the FOIL is O, so that would be the outside, so we go with the outside of each set. So the outside of this one is the big T, and the outside of this one is the little y. So we put the big T, and then you really want to make sure you make it look different, otherwise you'll get messed up when you start putting things together. So that's my little y. Now the inside letters are little t and big Y. And then the last, for the L, the last two, little t and little y. Now, those are the only possible combinations of sex cells or gametes that you can get. And so I'm just going to draw. This helps me remember that we are talking about gametes, so this would be like a sperm cell over here. Now that it doesn't matter if that's the egg, but for this one we're going to say that this is the sperm side, so this would be the male's possible sex cell gene combinations. Now since we're dealing with two of the same genotypes, the female over here has the same genotype, we're going to have the same gamete combinations. So we don't have to do FOIL again, we just write the same stuff on the top that we have on the side. So we have a big T and a big Y, then we have a big T and a little y, and then we have a little t and a big Y, and then we have a little t and a little y. So that's what we'll have across the top, just like on the side. So that part is done. Now all we have to do is combine our genes back together, because we are talking about sexual reproduction. So sperm meets egg, and the genes reform or recombine. So what we do here in this box is we go with big T, and we do another big T, and we have big Y and big Y. And we have big T and big T, and we have big Y and little y. Big T, little t, big Y, and big T, big Y, little y. That's the first set of possible combinations. Then we go down here, big T again, big Y, little y. We always want to put the big letter first if there's a choice. Big T, and little y, little y. Then we have big T and little t. And this does get kind of redundant, so if you are filling this out on your own, you can fast forward this video. You don't have to listen to me and watch me. You can just go right to when I'm done and check your work if you want. Alright, so that one, and then we have another big little, and then big little, and then we have little t and little t, big Y, and then little t and little t. And this does get kind of tedious when you do it, and you always want to double check as you're doing it. Sometimes it's hard for me to do this and talk. I might mess up the letter combos, but if you don't do it right now, then you'll find out in the end, especially with this kind of cross. I'll tell you why in a second. But these, when you do two heterozygote crosses, you always end up with the same ratio when you're all said and done. And then this one is a totally recessive individual. Okay, so that's our Punnett square, and everything looks like it came out right. So now you just have to ask, what's the question being asked? What has the teacher, or what's the problem that you're trying to find? A lot of times I will ask you for the phenotype ratio or the phenotypic ratio. Other times I'll ask you for the genotypic ratio. Well, remember, phenotype means physical. Alright, so I always remember pH starts both words. So phenotype equals the physical makeup, or what it actually looks like, what you can physically see. Alright, so according to our genes, what we have to figure out is the rule of dominance. If there's a big letter, then that's the trait that will show up. So for this one, we have two big T's, so it's tall, and two big Y's, so it's yellow. So we have a tall, yellow, and then what we want to figure out is how many of those we have. And what I do to make sure I don't mess up is I cross them off as I go. Alright, so I think the first one here is tall yellow. So we've got that one. That's done. So that's one. And I kind of do tally marks here to make sure I don't mess up. Here's another tall yellow. This might get a little bit messy here. I might have to do something different. Here's another tall yellow. Here's another tall yellow. Here's another tall yellow. So we're at 5 now. Here's, that's not a tall yellow, that's a tall short. I want to stick with what I'm looking for right now, and then we'll come back. Here's another tall yellow. And here is, that's not. Here's a tall yellow. Here's a tall yellow. That's not. There's one, there's a tall yellow. That's not, and that's not. So far we have 9. We have 9 tall yellows. And for future reference, whenever you do a heterozygote cross, you will always have 9 of the dominant phenotype. Alright, so now what do we have left? Let's get rid of maybe these ones that we already are using here, that we've already filled in. So that one, these are all gone. I'm going to shrink this here. These ones are gone. Gone. And that one's gone. Now, the next phenotype that we have is this one. It's tall and green. So we write tall, green. And we count out how many we have. This one is a tall green, so there's 1. That one's a tall green, so there's 2. And this one down here is a tall green, so there are 3. So we have 3 tall greens. So we can get rid of those. Get rid of some of this other mess that we have going on here. And that's that. So now the next phenotype that we have, we've got short in yellow. And there's one of those. And here's another one, short yellow, so there's 2. And short yellow, there's 3. So we have 3 of those. And then our last one, as you can probably figure out now, is the totally the homozygous recessive one that's left, and that would be the short green. All right, and we have one of those, and we cross that one off, and that leaves us with an empty Punnett square. So our phenotypic ratio then, on a heterozygous cross with complete dominance, our ratio will be 9 to 3 to 3 to 1. And that is always going to be your phenotypic ratio when you have a cross like this. So I hope this helps. This will help you guys solve the problem that you guys have for your weekly assignment list, which would be this one. There's a little backtracking and some things you have to do on this problem that makes it a little more difficult, but once you actually get the cross set up, it's no different than what we just did. So this is how we do our basic dihybrid cross. I hope this helps. Please ask any questions either through Moodle or send me an email, and I will hope to see you on Monday.